What if I told you that this single greatest discovery of the 21st century has the power to cure all genetic diseases, reshape our planet's food supply, and maybe even cure cancer? And what if all of this was found in something no bigger than a grain of dust? Because all this stuff isn't science fiction anymore. It's real. And it all started somewhere no one expected. Inside bacteria. It's called CRISPR-Cas9, and it might be the most powerful scientific tool we've ever discovered. But it didn't come from high-tech labs or supercomputers. It came from bacteria. In the late 1980s, scientists studying bacteria noticed something weird. Tiny repeating patterns. They didn't fit. They weren't part of the bacteria's genes, so what were they? For years, no one knew. Until one discovery flipped everything. Those repeating sequences were memories. Bacteria were remembering their enemies. When a virus attacked, the bacterium kept a tiny piece of the invader's DNA, like a mugshot, and it stored it inside its own genome. So if that same virus came back, the bacteria could recognize it instantly. Then call on an enzyme, Cas9, a molecular pair of scissors, to slice the virus's DNA to pieces. That's not just defense, that's precision. And precision is power. The way this tiny defense system works is elegant. When the virus attacks, the bacterium transcribes the stored mugshot DNA into a small strip of guide RNA. This RNA acts as a molecular GPS system. The Cas9 enzyme then latches onto this guide RNA. The RNA's job is to scan the cell's environment, seeking a perfect match to find the viral DNA sequence it carries. To ensure it is cutting the right enemy DNA, and not the bacterium's own code, Cas9 requires a second critical feature, a short sequence called the PAM site. Without that tiny flag, Cas9 won't cut. When it finds that matching invader DNA, the Cas9 enzyme locks on and with two separate active sites makes a double strand break, essentially snipping the viral DNA in two, permanently disabling it. This process is incredibly fast and unbelievably specific. It's the ultimate search and destroy function, allowing the bacteria to adapt and survive. Two scientists, Jennifer Doudna and Emmanuelle Charpentier, realized what this could mean. What if we could borrow that bacteria trick? And what if we could use it in any piece of DNA we wanted? Not destroy, but rewrite. By using a synthetic guide RNA, you could tell it exactly where to go in a genome, and it would make a perfect cut, letting us delete, replace, or repair genes. And in 2012, they did it. For the first time in history, DNA itself became editable, but an idea that powerful doesn't stay contained for very long.
Well, Health This Morning, the FDA appears ready to greenlight the first treatment using gene editing.
Jennifer Doudna
won the Nobel Prize in 2020. This is a huge moment in science and also in medicine.
Nobel Prize-winning CRISPR gene editing
I
technology.
invented a new technology. It's called CRISPR-Cas9.
Just 1 year later, Dr. Fen Zhang and his team proved that CRISPR-Cas9 could not only work in bacteria, but in human cells. That changed everything. The sudden idea that a child could be born with a genetic disorder and have their DNA edited and live a completely normal life. It was a complete game changer. Researchers used CRISPR to develop rice and wheat resistant to disease and drought. The future of the global food supply suddenly looked secure. The first clinical trials were approved, scientists began to develop therapies to turn a patient's own immune cells into cancer-fighting assassins.
The first time scientists try editing a gene inside a body.
Of an adult patient for the first time to treat a genetic form of blindness. It was a cascade, a scientific shockwave. What used to be science fiction was now a global research protocol, promising cures, better food, and unimaginable breakthroughs in medical and scientific fields. But power without restraint is always dangerous.
Chinese scientist helped create the world's first genetically edited babies. He is going to jail.
Twin girls, making them resistant to HIV. The world's reaction? Outrage. Because no one knew the consequences. Had he protected them or doomed them? Had he fixed a disease or created a mutation that would echo for generations? He was sentenced to 3 years in prison. That day, the world was reminded the power to repair comes with the heaviest of human responsibilities. But when used wisely, that same power has already saved lives. In 2023, doctors used CRISPR to cure sickle cell disease, a painful, lifelong blood disorder. They took a patient's own cells, edited them, and gave them back stronger. Within months, the disease was gone. People who had spent their entire lives in hospitals were now healthy. And just 1 year later, CRISPR saved a baby. Born with a rare genetic condition that would have been fatal, his doctors edited his genes, and he recovered. It's hard to overstate how wild this is. We're no longer just treating symptoms, we're fixing the disorder itself. So, what's next? CRISPR could one day erase hundreds of genetic diseases. In fact, this tool is already improving. Scientists are moving on from the cut and paste of Cas9, developing new systems like base editing and prime editing that can precisely swap a single DNA letter, no cutting required. But some scientists want to go further. What if we could edit aging itself? Could we one day repair the DNA damage that makes us old? A longer health span, not just lifespan. It sounds impossible, but so did editing DNA. And that's what makes this story so profound. The crazy thing is, this revolutionary power emerged from a minuscule defense mechanism inside a single-celled bacteria. For decades, those strange, same repeating patterns sat undiscovered, dismissed as scientific noise. But that one small, random thing, once understood, changed the course of human destiny. It really makes you wonder, what other astonishing possibilities are hiding in plain sight, just waiting for the right question to be asked? So, if you want to rewrite history, then subscribe, and I'll catch you in the next one.